Hello, everybody. Hello, world. Hello, Philippines. And hello, all countries and parts of the world. Welcome to my YT channel. This is again a singer Eternal Voice. So, this time I'm gonna give examples for enzymes. So, let's make a quick review of what is really an enzyme. Okay, an enzyme is basically a catalyst, the same principles with catalyst, but enzymes are mostly used in terms of the biology, you know, the physiological reaction inside our body, okay? And catalyst is applied mostly in the process, okay, to produce particular products, and we need to speed up the rate of the reaction, then we need to add a catalyst. But in biochemistry or reaction inside our body, okay, to produce different macromolecules, for example, nucleic acid, carbohydrates, okay, protein, sugars, like that, nucleic acids, DNA, RNA, like that. We need to have enzymes reaction inside our body, okay, to speed up the rate of this macromolecules, to produce these macromolecules, okay. So enzyme is a protein-based catalyst, okay, that catalyze the reaction and speed up the rate of the reaction, okay, like that. So it's gonna be like this. I'll illustrate like this. This is our enzyme, okay. And this is our substrate. The substrate and enzyme will react together in the activity site. So this is our activity site, okay, and then there's substrate will go there, and in a perfect clipping, meaning reaction to this, shape of an enzyme. It's gonna be like this. Enzyme, so this is gonna be like this, okay, as our end product. So this is our final product or our result upon the reaction of the substrate to this enzyme in the activity site. So this one will go to the activity site perfectly. So this is the principle of an enzyme, okay. So enzyme are protein-based catalyst, okay, and then there are a lot of exemptions also to that. We have the RNA catalytic substances, for example, ribozymes, pepsin, trypsin. Those also are enzymes that does not end in ASE, okay, but majority of the enzymes end in ASE. So I'll give you an examples, okay. Enzymes in the salivary gland, okay. So we have the enzyme, substrate, the products, like this. Like, okay, we made a table showing the relationship between the enzyme, the substrate, and the products. Here, I'll be talking the digestive system. So this is salivary gland. So meaning to say the products of digestion actually are the products of our enzymes and substrate here. So if our enzyme is, let's say, chaline, see chaline doesn't end also with ASA, but this is an enzyme, right? Chaline or amylase, salivary amylase. Okay. Chaline. Our enzyme is chaline or salivary amylase. Our substrate will be, okay, the starch, and the products of digestion will be maltose. Okay. Okay, products of digestion will be maltose. Starch is a polysaccharide. Okay, that is composed of 4 beta glucose monomers lying glycosidic linkage. 4 beta glucoses monomers glycosidic linkages. This starch. Okay, multiple simple sugars added together, that is a starch, and then it will produce maltose. So maltose meaning to say 2 double sugars, glucose plus glucose. Okay, like that. So maltose undergo the decomposition reaction of 2 glucoses or double sugars, glucose plus glucose. Okay, like that. But in here, the products of digestion is only maltose. Okay, like this. You can find the sugars, maltose, usually applied in the fermentation industry. Okay, the malt, MLT, in the production of wine, beer, whiskey, wine, like that. So the responsible raw materials for that there are maltose, or the sugars found in that fermentation process are maltose. Okay, and then it could be break down also to several glucoses afterwards. Okay, but since we're talking only of enzymes here, okay, like that. So the starch, okay, is our substrate, and our product of digestion are maltose. Another example will be, okay, we have the uh, gastric gland. In gastric gland, we have to divide it to 2 for the adults and I for the infant. Okay, for the adult, we have the enzyme is known as uh, pepsin. Okay, pepsin. For the adult, or I'll put capital letter A, pepsin is also an enzyme. This is for the gastric gland in the digestive system. Our enzyme is pepsin for the adult, and for the infant, baby infant, is renin. Okay. So our substrate for pepsin for adult, our substrate will be the protein. And our product of digestion will be peptone. Okay, like this. And for renin, our substrate is milk protein or carcinogen. For the baby infant, our substrate is milk protein and carcinogen. The products of digestion will be casein protein. Okay, like this. Okay, for the gastric gland, we have the capital A for adult, with the capital I for the infant. So the enzyme for the adult, okay, necessary to produce peptone is pepsin, and our substrate will be the protein. So, okay, the pepsin and protein will bind together, okay, and they will produce a peptone. And for the baby infant, we have our enzyme will be the milk protein and the carcinogen, and the product will be the this one, casein protein. And now we go now to the pancreas. Okay, pancreas, yes, pancreas. So we have the pancreatic amylase. Pancreatic amylase, okay. Again, our substrate will be the starch. And our product of digestion will be the maltose. And now we have the trypsin. Trypsin also doesn't end in ASE, but that is a enzyme in the pancreas. Okay, trypsin is our enzyme, and our substrate will be peptone and protein. So it will produce polypeptides. Okay, like this. Now we have the chymotrypsin. Chymotrypsin. The next will be chymotrypsin. This is our enzyme. Our substrate will be protein. And it will produce peptone, polypeptide, and di and tripeptides. For the chymotrypsin, our substrate is protein. It will produce polypeptides, the ditripeptides, and the peptone. Okay, now we have the pancreatic lipase. The pancreatic lipase will produce, our substrate will be fats or lipids. Okay, and it will produce glycerol and fatty acid. It's gonna be like this. Our enzyme will be pancreatic lipase. Our substrate will be the fats, and our products of digestion will be the glycerol and the fatty acids. So these are all the digestive system. Okay, but there is another one, the intestinal gland. Okay, like this. intestinal gland. So this intestinal gland enzymes are common, right? Because we can easily understand. Okay, for example, this is our intestinal gland, which are the large intestine and the small intestine. Intestinal gland. So our enzymes, a lot, I think four. We have the lactase, okay, the sucrase, okay, the maltase, and the amylase. Intestinal gland. As you can notice, there is also amylase enzyme. So these four are enzymes in the intestinal gland: lactase, sucrase, maltase, and amylase. We have also pancreatic amylase, right, in the pancreas, but this is also in the intestinal gland. So the amylase is very active. This also an enzyme in the salivary gland, in the pancreas, and the intestinal gland. Okay. So this is our enzymes, and our substrates will be for the lactose. Okay, lactase, our substrate will be lactose. Okay. So our products of digestion will be glucose and the lactose. So like this. Okay. Our substrate is lactose, and then our products of digestion will be glucose and the lactose. Glucose and the lactose are mono sugars or simple sugars. Okay, and then lactose is a disaccharide or an example of polysaccharide. Okay, like that. Lactose is a formula of carbon 12 H22O11, the same as sucrose, okay, and maltose also. But this glucose is carbon 6H12O6, the same with glucose, okay, C6H12O6, and the same also with dextrose. Dextrose is a sugar also, okay, that is being used in the patient during admittance in the hospital to give energy to the patient. Dextrose is also a sugar, carbon 6H12O6. Okay, like that. So lactose, maltose, and sucrose are disaccharides or polysaccharides, and the formula, general formula, is carbon 12H22O11, and this glucose, galactose, and fructose are monosugars or simple sugars, which is the formula of carbon 6H12O6, monosaccharides, or let us say simple sugars. It is very important we can determine which are examples of disaccharides, okay, monosaccharides, and polysaccharides, so that through that we can distinguish easily the general molecular formula. Okay, and then for the sucrase, we have sucrose is our substrate, and our products will be glucose and fructose. Okay, our enzyme will be sucrase, our substrate will be sucrose, our product of digestion will be glucose and fructose. Okay, fructose are sugars found in most citrus fruits. Okay, and glucose found mainly in carbohydrates, okay, in starchy foods, or in adenosine triphosphate, okay, molecule during phosphorylation reaction by adding phosphate group in the reaction of photosynthesis. Okay, remember that one, photosynthesis. So meaning to say the glucose from the adenosine triphosphate ATP plus oxygen gas will be equal to carbon dioxide plus water. Okay, so our source really is ATP, and ATP will proceed the complete reaction using the light energy of the sun. Okay, and at that time there will be a phosphorylation reaction by adding phosphate group to ATP, adenosine diphosphate, and then this we can obtain glucose from ATP to proceed for further reaction through the photosynthesis, and will produce carbon dioxide and water. Okay, so to make the plants and animals survive, okay, also the human being or any living organisms can survive because we need carbon dioxide as a complete cycle, right? We need to release the carbon dioxide as human beings, and then it will be received by the plants, okay, and the plants will give back again the oxygen to the human being, okay, plus the oxygen in the air surrounding or in the environment. Okay, two sources of oxygen that we intake: oxygen from the air in our surrounding and oxygen coming from the plants. Okay, something like that. This goes back with a cycle. Okay, like that. Okay, or it goes back in the Krebs cycle, something like that. If you can remember, right, in the glycolysis reaction. Charot. Okay, now we have the maltose. Our enzyme will be the maltose, and our products will be the glucose plus glucose. Okay, the maltase is our enzyme, the substrate is maltose, and our products of digestion will be glucose plus glucose. Okay, and then we have the amylase. Our substrate will be the fats, and our products of digestion will be the glycerol and fatty acids. Okay, our enzyme will be the amylase, our substrate will be the fats, and our products of digestion will be the glycerol plus the fatty acids. So it's in here that we need a fatty acid to digest all the fats in our intestinal gland in the small intestine and large intestine. Okay, I don't have to specify the formula, but as you go along the way and take down notes and research for the molecular formula, then you will learn a lot for this. Okay, this is only these are all examples of an enzyme in the digestive system. Okay, like that. These are all in the digestive system of a human being. Okay, we have the salivary gland, we have the gastric gland, the pancreas, and the intestinal gland. Okay, and then we have enzymes in the second column, substrate in the third column, and the products of digestion in the fourth column. So it depends on the application, because this is only for the digestive system. If this is not applied in the digestive system, I think there will be another products will be produced. But the purpose really is to speed up the rate of the reaction, to catalyze the rate of the reaction of the different biological substances inside our body, okay, to produce the products. Okay, most of the times the products will be the macromolecules. It could either be the protein, the nucleic acid, the carbohydrates, like that, amino acids, sugars, like that. What else? Hormones, okay, also macromolecules, hormones, the lipids, the fats, okay, like that. These are really the products. So most of these macromolecules products from the initial stage, which are the biological substances or our raw materials inside our body, I'm not talking about the process. And then we add an enzyme to catalyze the reaction, to speed up the reaction, to produce the desired products, which are the macromolecules that made us healthy and stronger every day. Okay, so because they are really the, the the substances that uh, speed up the production of these macromolecules to boost our immune system. Okay, health immune system to, to defend us against any diseases or any disease causing pathogens or any disease causing or foreign materials, infectious pathogens. Okay, so that our immune system will be strong. Okay, something like that. Okay, so those antioxidants, vitamins, minerals, nutrients that we take also needs the enzymes to proceed the complete reaction to produce this essential, let us say, biochemical substances that we need to survive in this world. Okay. Thank you so much for listening for this short live stream, and I hope you get a little bit of information. And then if you are not subscribing yet my channel, then you can subscribe, you can comment, you can like in the comment section down below, and then click the newest updates of Sotero. I am a senior eternal voice. This is again Engineer Sotero Nepomuceno Jr. Sapid Nepomuceno Jr. Okay, safety engineer uh, and a chemical engineer by profession. Thank you so much for watching. Bye bye. God bless everybody.